Hepatitis B is a disease that may lead to liver failure and cancer. This disease may lead to a trip to a hospital or a jaundice and rarely death. As you can see by this diagram, hepatitis B causes decay in the liver. However, the advancement of medicine has generated many treatments for this disease. The most prevalent and effective cure for this disease is the use of a recombinant hepatitis B vaccine. This vaccine is manufactured using recombinant DNA like the Piglo lab. It uses the hepatitis B surface antigen, which is the outer, you could say the envelope of the hepatitis B virus. These rings, the envelope of hepatitis B virus, this is the hepatitis B core antigen, the one that is harmful to the immune system. The significance of the hepatitis B surface antigen for developing the vaccine is that it can be used. That it's harmless, but it can be injected into the patient, and so that their body can form antibodies to combat the virus whenever one enters the immune system through an infection. The first step in making the vaccine. Is to extract the gene that codes for the hepatitis B surface antigen from the virus itself. This is done through killing the cell and salvaging the DNA and then purifying it to get rid of the contaminated particles in the solution. Restriction enzymes are then used to cut the DNA in the precise places needed, and then process DNA ligation performed. DNA ligation is when portions of the DNA, one from the host cell and one from the recipient cell, fuse through by DNA ligase. After that, the plasmid is then inserted into the yeast cell. The most common yeast cell is Saccharomyces cerevisiae, or baker's yeast. This yeast is common for letting bread rise when it's baking. As you can see, this is the bacteria of the yeast under a microscope. The yeast cell is then grown in a yeast extract medium full of sugar so that it can ferment. However, instead of fermenting acids, it ferments hepatitis B surface antigen. The cells are then extracted and washed. Afterwards, they are purified by column chromatography or diafiltration. Diafiltration is used to. Afterwards, the resulting vaccine can be used by patients to treat hepatitis B and develop an immunity for it. After the recombinant hepatitis B virus is so effective that people who take it actually show immunity to it for most likely the rest of their lives. The production of this vaccine is similar to the production of glowing bacteria from the Piglo lab in many ways. One way is that genetically modified plasmids were inserted into organisms to produce products that they otherwise have produced naturally. Another similarity is that a natural reaction from both processes was taken advantage of to produce the desired products. For example, fermentation by modified yeast cells created hepatitis B surface antigens instead of acids, and the regulatory gene controlling the enzymes that broke down arabinose instead made green fluorescent proteins. These two procedures show that recombinant DNA is useful in altering behavior of organisms to achieve the desired effect. In addition to the large number of hepatitis B surface antigens resulting from the insertion of modified plasmid into yeast cells, make the recombinant hepatitis B virus one of the most effective vaccines in dealing with the hepatitis B virus.